All right, go ahead and get started here. And if any stragglers join in, that'll be excellent. Thanks everybody for joining us today for Ramona's webinar on scaling functional imaging with parallelized microscopy. My name is Natalie Alvarez and I'm product director here at Ramona. I'll be your moderator for today's webinar. So before we jump in, I just want to cover a few quick housekeeping items. First, if you experience any issues throughout the broadcast, please feel free to put them in the chat function, and we'll work with you to address those as quickly as possible. Second, this presentation will be available on demand after today's live session. So if you miss any part of it and want to rewatch it or would like to share it with any colleagues, you'll be notified in a few days by email with that final recording. Third, we'll be taking time to answer questions at the end of the session. So hopefully everyone can see the Q&A function should be at the bottom of your screen, but we encourage you to submit questions as they come up throughout the talks, and we will address those at the end of the hour. And if there are any that we do not get to, we can follow up with you via email. As a final housekeeping item, our goal is to make this webinar both educational and engaging. So it's going to be a little bit different than other webinars you might have seen. We'll have some polls sprinkled throughout the session, and we'll offer Amazon gift cards as prizes to our most engaged participants, including those who answer our questions correctly and also ask thoughtful questions in our Q&A. So before we jump into the technology and talks, I want to just start with a question for context for you all and your current imaging challenges. So hopefully everyone can see the poll here that popped up on the screen, but we'd love to hear, what is your biggest pain point with live imaging today? All right, we see some answers coming in. I'll give a moment for everyone to respond. All right. Well, thank you everybody for your input here, and definitely stay tuned for some more pop quizzes as our meeting goes on. So this webinar will highlight how our technology can enable you to observe dozens of samples at high spatio-temporal resolution simultaneously. And we're excited to share two case studies from leading model organism and organoid researchers today. But before I introduce our speakers, I want to quickly introduce our technology at Ramona. Our goal at Ramona is to help you go from images to insights in a matter of seconds, not minutes or hours. And to do this, we've really created a paradigm shift in microscopy. So when we think about the evolution of microscopy over the past 150 plus years since its invention, really not much has changed. Existing optical microscopes remain largely confined to one objective lens for one human to peer through. And while automated motion and digital cameras have increased throughput, most state-of-the-art high-content imaging platforms continue to utilize a single objective that has to repeatedly step and scan across multiple fields of view. However, for large samples, that process is very slow. For live samples, it's tedious, and it's very difficult to capture dynamic changes happening in real time across the wells of a well plate. So enter the world's first gigapixel microscope. With a call to action from the NIH, our team set forth to break this trade-off that has traditionally existed between field of view and resolution, with an initial vision of examining freely swimming zebrafish with enough resolution to pick up neuronal activity. And the solution was to invent an imaging system that is fast for large samples and both easy to use and, importantly, non-destructive for precious living samples. So what we've done is essentially leverage the evolution of camera technology as it's reached smaller and smaller sizes for cell phones. And we packed those cameras densely together into an array. This multi-camera array microscope, or MCAM technology, enables the cameras to work synchronously to capture live biology. So the visual on the right demonstrates our Vireo systems array, and that provides a sense of how quickly we can scan a 96 well plate as compared to a traditional single objective system. So this MCAM technology powers two products today, our Kestrel and our Vireo imaging systems. The Kestrel is designed to be the world's most comprehensive imaging system for analyzing behavior, morphology, and fluorescence at video rates in model organisms. The Vireo was designed to be the world's fastest live cell imaging system. It's particularly well suited for quality control applications, high throughput drug screening, and real-time cellular dynamics. Over time, we've grown our product offerings beyond this hardware to include a detailed suite of analysis tools, and we've incorporated machine learning models to rapidly deliver quantitative insights at nearly real time along with your image data. So now I would like to introduce our two amazing speakers for today's webinar. And let me pull up this slide here with their background so you can see. Our first speaker is Dr. Franz Kuchling. He's a postdoctoral researcher at the Tufts Allen Center in Medford, Massachusetts, where he investigates the temporal characteristics of cellular adaptation with a particular focus on cancer treatments. He received his PhD in biology from Tufts University under the supervision of Dr. Michael Levin, where he established Volvox carteri as a model organism for studying a neural adaptation and information processing. Dr. Kuchling also holds a Master of Science in Physics with a specialization in biophysics from the University of Heidelberg and a Bachelor of Science in Physics from the Karlsruhe Institute of Technology. His academic journey includes a visiting fellowship at the Wellcome Trust Center for Neuroimaging in London with Professor Dr. Carl Friston. He has also conducted research at the Center for Organismal Studies in Heidelberg, the National Institute for Basic Biology in Okazaki, and the Centro Andaluz de Biología in Seville, gaining expertise in advanced microscopy, gene induction, and quantitative image analysis. Dr. Kuchling's research integrates biophysical, computational, and experimental approaches to understand pattern formation, adaptation, and morphogenesis in biological systems. He utilizes advanced imaging techniques and mathematical modeling to uncover the physical and informational principles underlying development and disease. Welcome, Dr. Kuchling. Looking forward to hearing from you. Our second speaker, Dr. Patrick Fortuna, is a Wyss Technology Development Fellow at the Wyss Institute at Harvard University, where he works in the George Church Lab, developing advanced stem cell-derived organoid systems to model human development and disease. His research integrates stem cell biology, synthetic biology, and tissue engineering with a focus on engineering complex multicellular structures and dissecting cell fate decisions using cutting-edge imaging, single-cell sequencing, and transcription factor-based programming. His current work centers on cardiovascular research, aiming to achieve in vivo-like maturity and greater tissue complexity modalities as such as vascularization. Dr. Fortuna earned his PhD in biomedical engineering from the Queensland University, where he developed novel 2D and 3D differentiation platforms to study neurodevelopmental disorders. And he also holds advanced degrees in biotechnology from the University of Queensland and the Technical University of Denmark. He has previously worked at MIT, the Technical University of Denmark, and Biogen, contributing to innovations in programmable organoids, metabolic engineering, biomanufacturing. Through his interdisciplinary work, Dr. Fortuna aims to create physiologically relevant in vitro human tissue models that accelerate the development of personalized therapies for cardiovascular, neurodegenerative, and reproductive disorders. Without further ado, I'm excited to hand it over to today's first speaker, Dr. Franz Kuchling, to describe how he's using our Kestrel system to study the behavior and learning of Volvox.
Hey everyone, nice to meet you everyone. Thank you, Natalie, for this kind introduction. I hope you can see my screen, that should be good. So today I'm going to talk to you about my PhD project, which was on uncertainty minimization and the adaptive photoresponse of Volvox. Basically taking principles from neuroscience and adapting them down to a very basic organism and see if we can learn something new. So kind of the question really underlying all of this is, what is the origin of cognition, which you can define in the Oxford University Press as the mental process of acquiring knowledge through thought, experience, and the senses. And that can be, just like in a PhD, pretty torturous. And so the kind of motivation for this work a little bit is this, or at least illustration, is this torture device that actually got used in medieval times. I've actually met people that have experienced this. It's a real thing, got used in East Germany. And it works by having water dropped on the top or the back of the head of the person being tortured. And after a while, these simple water droplets become really like heavy, like a hammer on your head. And the reason this is so torturous is because those droplets don't just come at a very paced stream, but they almost drop in like these randomized intervals because there's so much noise in the system. And so the idea is your body can't adapt to it. You cannot really desensitize. A more scientific experiment was done on rats by Wyss et al. In the 70s, where they put a rat in a little cage with an electrode attached to its tail, and there was a lever and a little a little speaker inside the cage. And so sometimes there'll be a little warning, an auditory warning inside the cage, and afterwards the rat gets shot. But if it pushed a lever, I think it's right here on the left, then it could actually turn off the electrical signal. So it demonstrated associative learning in this model organism. But the control experiment was actually more interesting for me because in one of the controls, the lever didn't actually work, but just the fact that it got that auditory signal and it got used to it and could therefore predict the electrical shock, the stress measured by ulceration was reduced. So even this mouse can apparently predict these shocks based on the signal, and that itself reduced something in its body. So it seemed to have basically adapted to it at least somehow. So from that, we can conclude that organisms with brains try to minimize uncertainty in the environment. And we want to figure out whether or not this is true for aneural organisms, so organisms without a brain. And so the foundation that on the mathematical sense that I base this work on is active inference, which is employed in neurosciences. The basic idea of this framework is that you have an active inference agent, like a human, that can sense the environment through its sensory states, like its eye, that transmits down these signals to the internal states, the brain, and that by itself can form its actions, like moving this eye, looking somewhere else, or just moving the body itself. And thereby it infers the external states from the environment. The actual mathematical formalism really doesn't have anything specific to your neurons. So it doesn't really matter if the neurons are fast or not. It's just about how these states are being updated from sensation to action. And that some kind of representation of the environment can happen inside. And so the argument we make is that this is true for a cell. We have a nucleus that encodes internal states, like its genetic states, expression. It can take actions by making new proteins, and it can therefore interact and predict this environment. And so what we want to show is that really, using this framework, the origins of cognition lie in cells, not in organisms, such as the photodidactic Volvox, which is an algae. So that's where we're applying this to, which is basically a two-cellular organism that gets used to study the transition to multicellularity. It has these reproductive cells on the inside and somatic cells on the outside that have a photoreceptor and two flagella that allow it to move. And so it can basically use flagella to create movement, or like to essentially swim. It pushes water around its surface and swims in the direction that it sees light. The reason I picked that organism is because it has a very measurable response to an external input, light, and it's just big enough to pick out with kind of low resolution microscopy. And so kind of this is like how they move in real life. You can see now where they get their name Volvox from the Latin word volvere. They revolve around, which is actually part of its adaptive capability to desensitize and resensitize it to light by always exposing a different part of its sphere. And so what we want to use this very unique but easily observable movement to kind of get at this active inference idea of uncertainty minimization. Basically, active inference is minimizing the uncertainty in your environment, predict it and act accordingly, which means that the more predictable stimulus, the better you can adapt to it, because it's easier to minimize that uncertainty from it. So the way that I approach this on an experimental level, so I put Volvox in these chambers and I would expose them with two different light pulse series, just two different LEDs at the end of the chamber. One, for example, would be this regular fixed interval frequency on, off, on, off. On the other side, it would have a more irregular randomized pulse series. And the idea is that, well, if it adapts more readily to a predictable stimuli, it should move more readily and adapt better to the fixed pulse sequence. Because, right, the randomized is basically the torture signal. And so I kind of gave you the big picture overview, and I'll now kind of focus a bit more on the nuts and bolts of how I started off on the manual counting system in the beginning of my PhD, and then decided that if we really want to get at this question in a high throughput way, we have to start imaging, and how the MCAM Kestrel really helped me with that. So for the manual counting, essentially, I 3D printed the setup, glued some glass lights on the side of it, had the LEDs all wired individually on the outsides, and controlled these with a Raspberry Pi. And then I would, in these middle sections, I would put little PDMS inserts that would just confine Volvox on there. So I would put these Volvox colonies in the middle of the chamber. I would move these stoppers and then watch them move as I basically turn on these different light patterns on the two different ends. And after 30 minutes, I would essentially put in new stoppers, dividing my chamber in different segments, collecting each segment into different welds of just a weld plate. And then we would do manual counting, especially Susie Zaki, my student, who unfortunately had to do a lot of this initial manual counting work. And then in the end, it did work. We did actually get a bias. So in percent of the average or the axis, we did see a more significant bias towards the fixed 1 Hz side rather than the random one. So they did seem to adapt better to that fixed pattern stimulus. But then we wanted basically to really get more information of the movement, the rotation, do some other experiments with different drugs in the media, and different light patterns. And then we just eventually decided that we can't just have Susie work 24/7 on this. After initially trying out some cameras myself in the lab, just like a single Basel camera, I decided that it's time to scale up. And so we got this MCAM Kestrel, which essentially has all these cameras on the top, like you just saw in Nali's presentation. And then I custom made this setup in the stimulation setup on the bottom here. We essentially have individually addressable LEDs on each end of these chambers. So we have 24 sections, one for each camera, divided into 2 chambers. That's 48 chambers at 20 Volvox each. That means I have now 960 Volvox that I can simultaneously image. And the imaging basically happens because I'm using those blue lights from these LEDs as an excitation length. And I noticed when I did this in like a spectral microscopy setting, I noticed that they have a autofluorescent far shifted like a Psi5 channel. So we just hooked up a Psi5 filter just below the cameras, and we got a very clean signal of the Volvox that looked like this, essentially, where you have all these chambers and you see those very small green dots here in the inside. So then all these LEDs were controlled by Arduino boards. And so the question was, how can we parallelize and synchronize and really integrate all of this into the image acquisition? So the beauty of, for me at least, of the MCAM system, that you do have a very nice graphical interface that does pretty much everything that you can do otherwise, but it also has it's built on Python, and you have a direct Python interface with it as well. So I'll just kind of walk you through like the main parts of the code that I think make this very consistent for me and reliable and customizable. You just, in the beginning of the code, you select which cameras you want to use, like just the Y coordinates and the X coordinates of your X and Y of your cameras, how long you want to image for. And then you can also select just like the cropping size. So you can basically reduce, and also kind of what your frame rate and what your actual pixel size your binning is. So you can do like a lot of the pre-filtering on the imaging acquisition itself, like less area to image and less temporal or spatial resolution, and thereby reduce kind of your data acquisition. And then we just hook this whole thing up to the Arduino, so that there's just like a little Python interface that you can use to open up a serial connection. You use USB cable with Arduino, and we control the Arduinos. So we tell it to run the program, and then we do the actual acquisition. You do have to allocate the memory first. This is important because basically, imagine you have 24 cameras, you're doing this high resolution. Everything has to be saved in the RAM, because otherwise, because you have such a high frame rate, at least for me, I had 4 frames per second. So that gets you about quite a bit of data. So I just upgrade the RAM and had now 500 gigabytes of RAM into that system, and then I could record 30 minutes at high resolution for 3 frames per second, no problem. So this just makes sure that this is actually really safe beforehand. So basically, if you don't have that RAM, it will tell you beforehand and not stop like halfway through, which would be awful. So this is very nice, robust. Yeah, and then you start the acquisition. For like a 5-minute video of my essentially a quarter of max resolution, which is still pretty high, you get about 9.6 gigabytes of data, which I don't think is that terrible. It's manageable. And then you just start the acquisition and watch it go. And so that's what it actually looks like. So this is what you're seeing right now is 24 of these chambers split up in 2 individual ones. The white dots are the Volvox, they're dancing around, and the flickering is basically all the lights right, of the different patterns that are programmed into this. And then if you zoom into one, we can do the analysis and we really get individual tracks. So we can really see like how one Volvox moved over time. We can measure the speeds, position, all kinds of stuff. And what's nice is that the MCAM actually says it as like a Python intrinsic NC file, which you can then immediately do all your analysis within Python. So there's no exporting need. You can go straight to your analysis software and go from there. So that's nice. Not only does it save you time, but also when you then export videos and everything, that's more data that you have to save and lose and manage. So this saves all that. And then to just kind of like give you actual like a more high resolution video of how this is really happening. So for example, here on the left, we have this anti-phase pattern where you can just watch the Volvox go up as one light is turned on, and then that light is turned on off, and the other side is turned on, and they follow this down. I can watch this forever. It's very therapeutic. Less therapeutic is this random disco lights here, where you compare the fixed and the random pattern. But luckily, I'm not counting these manually anymore. I'm just having my Python program analyze this, and so I'll just basically see how many Volvox are present on each side. And you can do this for different frequencies as well. And then what we actually saw is that indeed there was a bias, as we've seen before in the manual setup, towards the fixed, regular, predictable pattern as opposed to the random side. And this was true for 2 different species of Volvox, carteri and Oreos. And that is really nice because that means it's not, not super specific about the slight genetic differences of the frequency response inside the system. And even more supported by the second result, which is that even though they both had this preference towards the more predictable pattern, they had different biases, a lack of bias and a bias towards a higher frequency of 2 hertz for Volvox Oreo species. So even though they have slightly different frequency responses, they both do this in my interpretation and threat minimization. So main takeaways are that active inference predicted an annual organism behavior based on uncertainty minimization that was basically unforeseen and quite surprising at this lower level. And also that because of this uncertainty minimization, it really means that Volvox have a low-level capability of predicting the environment. And so some broader impacts here is that we can use active inference to prevent intervention tools for medical and ecological interventions. Basic idea is like, well, if you now you not only have targets for drugs, for example, or for light stimuli, but you can also vary your stimuli, your drugs or whatever in temporal space, and you get a whole new access of intervention tools that otherwise you wouldn't have predicted. And so as Natalie mentioned earlier, right, my current focus is on chemotherapy, which is actually what I want to do in the beginning anyway, which is really apply this to something meaningful. And so right now what I'm trying to do is see if we can design temporal simulation patterns that will basically cause a reduced resistance of the cancer cells to the drugs and make the drugs more effective at actually killing it. And then we're also doing some further testing right now of the ability of Volvox to discern different patterns, which is also quite interesting and really and important for the idea of designing the novel intervention tools, of understanding really how fine-tuned is this organism actually at being able to differentiate different patterns, which the ability by itself was kind of unpredicted beforehand. So I do want to say thank you to especially my team of students that made a lot of, especially the earlier work possible, without which we never would have kicked this off anyway. And my mentor, Mike Levin, and Carl Friston, who I spent this time and learned the active inference framework with my colleagues. And a special shout out to my Ramona goat, Mark Harfush, because he's essentially the one that helps you if you have any issues with the system, especially with the Python, but also just having it run on the Linux system. There was one amazing story where I really just, the system wouldn't set up, and I just called Mark, and he was actually driving somewhere on the highway, and he just kind of like a pilot that, like, imagine like you're on a plane, the pilot is dead, and you have to fly the airplane and just press the buttons. You have no idea what exactly what happened, and it just after 5 minutes the problem was solved. So really thanks. I really appreciate all your hard work. Yeah, and then thank you for your attention. Oops.
Fantastic. Thank you, Franz, for your presentation and for being an early adopter of the Kestrel system for this fascinating application. I encourage everyone to please ask any questions in the Q&A function, and we'll answer as many of those as possible at the end of the session. Before we jump into Patrick Fortuna's talk, it's time for a quick poll to see if you were paying attention to Franz's talk. And again, this is an opportunity to earn a gift card, so encourage you to participate here. The question is, how many Volvox did Franz image simultaneously on the Kestrel? We'll give everyone a moment to answer that here. We've got a few responses here. All right, last chance to answer here, and then we'll show the correct answer. All right, so the correct answer was 960. Really an impressive sample size to have 20 Volvox in each of those 48 chambers in his plate format. So now I'm going to hand it over to Patrick for him to start his presentation.
Thanks, Natalie. Hope everyone can hear me. Thanks everyone for joining today, and Franz, that was a great talk. So today I'd like to talk about the Vireo system and how I've been using that to analyze cardiac organoids. So I'm a Wyss Technology Development Fellow at the Wyss Institute at Harvard University. Right, here we go. So first of all, how do we make organoids? Well, we need to start with stem cells. Traditionally, people have been generating embryonic stem cells. These are cells that were isolated from the inner cell mass of pre-implantation blastocysts. After 2006, though, a lot of people have been using induced pluripotent stem cells. These were made by introducing the 4 Yamanaka factors into somatic cells, which in turn you could reprogram back into a pluripotent state. So over the last 15 to 20 years, people have come up with these really beautiful protocols of how to generate tissue-specific organoids across the 3 germ layers. So we've become really good at making these, but now we're kind of running to the issue that we really need to try and better functionalize these to mimic what happens at an adult in vivo like stage. So what have people been trying to do to accomplish that? This isn't an exhaustive list, but a lot of people have been trying to introduce things like vascularization, trying to recreate the interaction between the 3 germ layers, trying to figure out how to mature these tissues faster, doing co-cultures across different cell types, and also trying to spatio-temporally control signals that happens. So specifically within cardiac organoids, people have been looking to either grow these in different biomaterials. So if you take cardiomyocytes, grow them in different biomaterials, or do the introduction of different cell types, because obviously your heart isn't just made up of one cell type. These kind of aspects together, people like to look at electrophysiological functions. And then you get deeper into more complex systems like generating cardiac organoids, where you're combining these different cell types together with different hydrogels. And also, if you want to better mimic biochemical, biophysical cues, people have also been using hard-on-a-chip devices. So what's nice across all these different paradigms is that people have been using these to better study disease modeling. This is where something like iPSCs are really handy because you can generate cell lines that have a background disease within it. You can look at drug testing. This is where maturation becomes really important because if you're able to really mimic what happens in an adult-like state, then you'll better, more faithfully reproduce what happens when you're adding these drugs and so on. So, but the one thing that really becomes an issue is that when you're trying to analyze huge amounts of these organoids, you run into the issue that you can't do this fast enough or in a high throughput manner. So this is where the Vireo system has been really handy. So previously, this is just a small example of what a differentiation protocol could look like. I'm not going to go into huge detail, but the main gist is people use protocols where you do temporal modulation of Wnt signaling. You could use small molecules, for example, like higher and XAV. When it comes to the functional analysis, though, something like when you're trying to image, though, you could run into the issue of long experimental setups. So you're imaging individual wells. When it comes to doing 96 wells, that can take quite a long time. You got to program your positions. And then when you're actually acquiring your data, you need to have integration across your data acquisition and analysis, because it's not always the case that how you're acquiring your data is going to have an end-to-end method to actually analyze it straight away. So a lot of people either do custom software, or in this case, you have to align your data formats in order to use that in these third-party packages. And one of the big problems as well happens is that you might find a third-party software that you really like to use, but then you run into the issue that a lot of these aren't maintained after they've been published. So you might find a really handy tool that you're using, and then over time you find that this starts to break down. That can be a big issue if you want to maintain consistency of how you're analyzing your data. So with the Vireo system, what's been really great is here, for example, I'm just showing a cutoff section of my 96-well plate. This isn't the whole 96-well plate. Here I'm imaging in bright field. I'm using a cell line you can purchase from Coriel. It's got a MLY7 GFP reporter in it. So it's nice, you can test how well did your differentiation happen, because if you image in GFP, you can see if this marker is actually present. Here I'm showing, for example, what happens when you're recording at 45 frames per second. So you can image the entire plate in under 2 minutes. Like Natalie said at the beginning, it's 24 cameras that image simultaneously. So it only moves 4 times to image your entire 96-well plate. So here I'm checking, what happened in my differentiation. Was I able to make these into cardiomyocytes or cardiac organoids in this case? But what really becomes powerful in the system as well is that you start to use these inbuilt analyses within the system. So here you can perform segmentation, and it can directly measure the sizes of your organoids. Here is a video of what this organoid looks like at day 9. I must say that this specific video wasn't taken with the Vireo system. This was just with the Echo microscope in the TC lab. But with the segmentation, it's really fast in the sense that you can perform your QC pretty much as you're imaging. So here's just a brief example of 10 random organoids that I picked over the first 4 days of induction. So you can track your measurements, the sizes of your organoids as they're progressing during differentiation. So like I said, you have these inbuilt functions or analyses packages within the system. But what happens when you want to do actual functional analysis? So recently, with Clay, one of the software engineers at Ramona, they released a white paper of doing calcium transient analysis. So you can check out the website because they recently published that. Here they just showed, and we've used this within these cardiac organoids, of measuring the calcium transient, so getting a normalized fluorescent change. In my case, though, sometimes I'm working with these reporter lines, so imaging within the green channel that isn't possible. So what you can also do is just do thresholding and look at the physical the physical movement, the changes that's happening as these organoids are contracting. So here, for example, we're measuring what happens in the pretreatment organoids when I added a drug like verapamil. This is a calcium channel blocker. As I increased concentration of the drug to 1 micromolar, for example, we can see that now these pretty much have stopped contracting. Another example of adding a drug, a lot of people like to use this as a control to see what happens with their cardiac tissue, is isoproterenol or isoprenaline. This is a beta adrenergic agonist. So when adding to 10 micromolar, you can see that we've almost double the contractions or measuring almost double contractions within these cardiac organoids. So with clay, we've been looking at ways of how we can increase like what sort of analyses we can do within these. So we've been looking at things like contraction time and relaxation time, so systole and diastole parameters. And this is now built in within the Vireo. So after you've acquired your videos in Brightfield, you can use one of the software options, like, or the analyses options directly in the software to show you, what's the contraction time, relaxation time, heart rate. So this is the same organoid that was treated with, before it was treated with isoproterenol. And then after adding 10 micromolar of that, we can see now that, we're almost at this double effect. And now you can get this nice readout of what is the beats per minute and how did the contraction and relaxation time change. Now this becomes pretty powerful when you're screening a large amount of drugs over a large amount of organoids. So that kind of leads me into one thing that can, be a bit scary or hard to deal with for people is data storage. So data storage and management can become burdensome. One thing that's a huge benefit in working with this, having the end-to-end processing included in the system, is that you're taking the compressed data files directly into analysis and then getting your output of what happened with these your organoids, depending on what treatments you have. So that saves a lot of time, both in terms of how you're storing your data, but what you're doing with it afterwards. So there might be situations where you need specific custom software because it might not be included within the Vireo system, but the Ramona team have been very open to working with us to figure out how they can include these different analyses within the software directly. So that's been a huge help. So that kind of brings me back to this. What are the pros of using this? So faster experimental setup, one, because you don't need to pre-program your positions. It's directly imaging like a full 96-well plate in under 2 minutes, which is super fast and handy because you can literally treat your organoids, put them into the plate reader, and then image across all of that, either just still images or videos. The output of the data files, whether you're using the raw or the processed ones, they're all standard format data files. So even if you want to then put this into your own analysis software, whether you've got a custom code in MATLAB or a macro in Fiji, then all of those are compatible to use across those. So this has significantly increased how much and how fast you can analyze your your organoids. I must say I've been primarily just working with the 4x objective Vireo system. They also have a 10x one, which if you're working with 2D cultures, you can probably get to single cell resolution if you're looking at things like calcium imaging or tracking movements, for example. So again, data storage could become an issue if you're doing really long time course acquisitions and so on and across different channels, because you can record in green and red fluorescent channels and in bright field. But so far, processing the data as you acquire it has definitely taken away a lot of that burden. So me personally, what I'd like to do moving forward is start to do make a lot of like large perturbation screens, both 2D and 3D, because you can also image in 3D 4-well plates within the system. This makes it really convenient if you want to do large screens specifically. And another thing I'd like to mention is that we're looking at assessing existing engineered heart tissue devices that are on the market, because a lot of teams also develop these and measure things like active force. So the Ramona team is looking at potentially trying to include that within the analysis package, like in the software itself. So that's me and what I've been doing with cardiac organoids specifically. I'd like to thank the Church Lab. We're a very big lab. You can see here in the picture from last year. Within the Church Lab specifically, Jenny and Esme that help, make the team run super smooth and efficient. The funding buddy, and then a special mention to Professor James Hudson from QIMR in Australia, who got me interested in cardiac organoids from the beginning. And the Ramona team, Aurelian, Clay, and Natalie, just been amazing at getting me set up, getting me up to speed with using the system, and Clay, always coming up with ways of how to analyze the data better. So thanks everyone for listening today.
Great. Thank you so much, Patrick, for that exciting presentation. And it's been a pleasure collaborating with both you and Franz. So now it's time for a quick pop quiz here. I'd like to ask everyone about the throughput that Patrick saw on our Vireo system. So how many cardiac organoids did he image within 1 minute on the Vireo? And we'll give everyone about a minute to respond to this question. All right, last chance to enter a response here. Okay, so the correct answer is 96. As this system has 24 cameras, we can record live video of 24 wells synchronously, but then we can quickly step and scan 4 times to image 96 wells within a minute, and Patrick has 1 cardiac organoid per well of his 96 well plate. So now it's time to jump into our Q&A session here. Encourage anyone to enter any last minute questions here and we'll address them now. We have a couple of questions that have come in. And first, I'd like to pose a question to both of our speakers today. In terms of interacting with Ramona's hardware and software, how easy was it to learn and use? Franz, do you want to kick us off?
Sure. I think the graphical interface and actually using the microscope was very easy to learn. Definitely faster than other systems. The whole Python interface, which again you don't need to use unless you have specific needs. As far as I know, mine was basically the integration with the Arduino control. That took a bit longer, but also wasn't a big deal. So I think this was pretty fast. I mean, you can base out experiments within a day or two, I would say.
Yeah, I definitely echo that sentiment. It's a super easy interface to use. You can basically anyone can just jump on a few learning rounds and you can set up a whole experiment pretty easily. Clay's also been great at generating some Python Jupyter notebooks that you can analyze your software, your data afterwards, but most of it is actually included in the system itself. So you don't even need to do that, kind of like what Fran said. You don't need to specifically know how to work in Python if you don't already. But yeah, it's super easy to use.
Great, great. So as a follow-up question to that, we have a question from Ian. He's interested in the hardware control. How does it work with synchronizing to external hardware components? Is it possible to make reactive events to biology, such as image analysis, trigger something like a fluidic pump kicking on? And I think I can answer that quickly from my perspective at Ramona. Franz was a very early user of our technology, and so we developed that Python code for him. And now we've developed a very user-friendly web-based API that provides the ability for our clients to use their image analysis and capture and give you results at every time point, for example, in a time lapse. And we can integrate external hardware like LED illumination in a closed loop configuration as Franz needed to do. We've also integrated with robotic scheduling softwares such as Thermo Fisher and Biosera's Green Button Go. So we can work with liquid handlers and automated dispensers, and we can provide those data streams to trigger actions. So Ian would love to work with you on that project. And Franz and Patrick, if you have anything to chime in on that, feel free.
Yeah, I'm not familiar with that new tool. I suspect that the only thing that you have to watch out for is basically your frame rate. So I don't know how. I have to imagine that you can correct me, but there's some limiting factor, like where we just if you go too fast, it won't be able to image, like to analyze in live every, 10 frames per second. But it's possible for sure. It just probably depends on exactly what your frame rate is that you need.
Right, exactly. We have a couple other questions from Ian as well. So he specifically was asking in response to Franz's talk, have you considered writing to ZAR with OME ZAR? I think there would be favorable write-up speed and cloud access benefits.
I have not, and I can also answer the second question there about the X-array. So that's basically MCAM, the whole Python package and the documentation they give you. They come like natively with X-array. So that's basically you have like an mcam.data function that essentially opens your data and that's based on X-array. And so I stuck with that because my software, the NELS that I did, worked just fine with that. I have not worked with Omizar. I assume you're right that from what I quickly Googled on this, that sounds like it's a bit faster and better compression, but given that everything works fast enough for me, I didn't bother. But it's probably a better question for Mono Optics to see whether or not they are thinking about Omizar or not.
Yes, we can definitely follow up with more information on that. All right, another question here. Do we have a plan to expand our business to the Asian market? Who is our channel partner in Korea? So thank you, Dennis, for that question. From a business perspective, we actually do have systems placed in several countries in Asia, including Taiwan and South Korea. I would have to get back to you on our specific channel partner. I know we have a system at a university there, so we can follow up from a sales perspective on that note. But we're always looking to expand internationally. There are some regulatory requirements, obviously, with regards to safety that we want to make sure that we comply with in terms of evaluating our system. But we are looking to place a system soon in Singapore, for example, and we've had interest from other companies as well in Japan. So happy to discuss this further with you. All right. I think that's all of our questions for now. I'm going to move on to a final poll here. Just curious for everyone to share which system they think might be the best fit for your research based on today's presentation. And there's no right answer here, so just feel free to answer. We'll give everyone a moment here to respond. You'll see some responses ticking up here, so we'll give a few more seconds. All right. Well, thank you so much, everyone, for your time today. This concludes our presentation. I encourage you to reach out to us at info@ramonaoptics.com or visit us on our website to learn more about our technology and book a demo. As a reminder, this webinar will be available live on demand, and you'll receive an email in your inbox in the coming days with the link. We'll also follow up once we tally our responses for our polls, and we'll reach out if you receive a prize. I think we have some former prize winners here on our webinar today. And then I wanted to let everyone know when exiting today's webinar, you will receive a quick survey. I'm leaving a few minutes here at the end of our session and hope you can take a moment to share your feedback on the webinar so that we can continually improve our programming. Thank you so much for spending the hour with us, and we look forward to being in touch. Take care.